All right, for the last bit of material for the first module of Anatomy 1, I'm going to focus on what is Chapter 3 in your textbooks. Cellular biology, cytology is the study of cells. I'll probably end up having to split this up in a couple different lectures. I don't know. We'll see how it goes. Now, in terms of human history, we haven't really known about cells for a very long time. It's only been a few hundred years that we have even known that there are cells. It wasn't until our technology developed enough for us to be able to make finely ground pieces of magnifying glasses and lenses and put them together in something that let us see small enough to where we started to realize, hey, there are things, there are little units that make up plants and animals and things swimming around in water. And so it wasn't until, there we go, it wasn't until about the late 1600s that we were doing this kind of stuff. Robert Hooke is one of the guys who gets credit for doing some of the first major discoveries there. Robert Hooke and Antony von Leeuwenhoek. Robert Hooke coined the term cells when he described plant little compartmentalized structures in plant tissue with this basic type of microscope that you see in the picture there. But even after that, it was still a couple of hundred years of, maybe 150 years of development and learning and experimentation and science before we were able to start putting ideas together and truly understand what is, at least the basics of what's going on. In the 1830s, some scientists, Schleiden and Schwann, they put these ideas together and they came up with what we still use today, and we call it our modern cell theory of life. They said that all living things are made of cells, that cells can only come from other cells. They don't just magically appear with spontaneous generation. And that, so what did I say? All living things are made of cells. Cells have to come from other cells, and that the cell is the basic unit of life. If it's smaller than a cell, if it's less than a cell, then it's not really alive. Sorry, viruses, you don't quite make the cut. And today we know, and you guys will see, that cells come in many different shapes and sizes. It really just depends on their function and what they need to be doing. We'll see many of these throughout especially Module 2, as we start seeing the tissues, the microscopic views of the parts of our bodies that have various functions for life. Regardless of the type of cell that we're dealing with, there are some characteristics that all of our cells have in common. That's what I'm going to focus on for this last lecture or two of Module 1. So here's a very basic diagram of a cell, very simplified. All cells have three main things, all of our cells. They have a membrane, a plasma membrane that's forming kind of a container for everything. All cells have cytoplasm with stuff, that's the guts of the cell, cytoskeleton, organelles, things like that. And all cells have a nucleus with genetic material for controlling what the cell does. We're going to take a closer look at each one of these components, characteristics, maybe that's a better word, of our cells, starting with the cell membrane. And actually, the membrane for us in anatomy, the membrane is where we'll end up spending most of our time, because the membrane is so important for understanding the physiology of what goes on in some of the things like muscles and parts of our nervous system later in this semester. So the plasma membrane is what we're going to focus on. The plasma membrane, it does act as a container, a wrapper. It separates the insides from the external environment, intracellular fluid from extracellular fluid. So it acts as a barrier. We got to keep some things out. We have to keep our organelles from just floating away, right? So we need a little bit of a barrier or wrapper. But the plasma membrane is also dynamic in nature. It has to allow for the transport of stuff into and out of the cell, specific things that the cell wants or needs to get rid of. Right? So the way the plasma membrane is set up, it's called the fluid mosaic model. It says that the plasma membrane is a phospholipid bilayer, a double layer, with proteins embedded into it that are always shifting around. It's fluid in nature, and it's a lot of different stuff working together, so kind of a mosaic. That's why they call it the fluid mosaic model. If we're going to understand the basics of a plasma membrane, I'm going to draw some things. It's a layer. I'm going to draw a layer of phospholipids. Phospholipids, like what you saw in the chemistry information, to me, phospholipids look kind of like jellyfish, maybe. So here's a phospholipid. But it's a layer of phospholipids. I'm going to draw a bunch of them, and I'm going to get kind of lazy. There we go. And it's not just a single layer, it's a bilayer. And the other ones are upside down. And then there are proteins. And more phospholipids. All over the place. I told you I was getting lazy. There we go. Close enough. Yeah, that works. Phospholipids naturally arrange themselves in this pattern. This part likes being next to water. It's hydrophilic. Hydrophilic. The tails, that's the lipid part, the fatty acid parts. These parts are, let's draw it, hydrophobic. I'm going to run out of space. Hydrophobic. They hate water. So the parts that hate water, they're attracted to each other and line up this way. While the hydrophilic parts like water, they're okay with facing towards the extracellular fluid and the intracellular fluid. The inside and the outside is just water with stuff dissolved in it. So they naturally line themselves up that way. That reduces the amount of work that the cell has to do. So here's a better diagram of that phospholipid bilayer. I mean, my diagram, my drawing is pretty, but here's a nicer one. It doesn't have any proteins. What about the proteins? Here's a more detailed version right there with a big old channel protein right here, transmembrane protein that goes all the way across from one side of the membrane to the other. So these integral proteins are built into the lipid bilayer. Some of the integral proteins are transmembrane. They go all the way across. And most of the time they involve transport in some way, like this one would act as a passageway, letting stuff go into or out of the cell through that opening. Sometimes they'll have a little receptor here on the surface that will recognize when certain molecules fit down into that socket just right to relay a signal, to carry a signal on from outside of the cell. So that would be something like a protein or some other molecule that's carrying a signal. That's a receptor. Peripheral proteins, there aren't any in this diagram, but they would be attached to those integral proteins, and they would be helping the cell to change its shape or link up together to another cell that it's next to, that kind of thing. Glycolipids and glycoproteins help to coat the outer surface of the membrane. That's these stringy things that you see in the diagram. They'll act as ID markers for the cells. So our cells can recognize each other, and our immune system won't target them and try to destroy them along the way. So those are the main components of a plasma membrane: phospholipids, proteins, and then these glyco things, glycolipids and glycoproteins. You want to be familiar enough with this setup of the plasma membrane, enough to recognize those components if you saw a diagram of them on a test. Sometimes they do make it onto the test, sometimes not, it just depends. I just mentioned that sometimes cells need to link up together, and we call those membrane junctions when those proteins help cells to do that. There are 3 types of membrane junctions that I want you to know about. First of all, tight junctions help to form an impermeable seal that keeps things from passing between the cells. If I'm ever in the classroom, I usually draw it out like this. Here's a few cells right next to each other. Nucleus. And so if we're dealing with tight junctions, we'd be forming a nice tight seal that wouldn't let anything get in between those cells. In your digestive tract, we don't want the acidic stomach juice leaking out in between the cells. We don't want the things that we're trying to digest or the other enzymes leaking out. So we have a lot of tight junctions to seal everything in. Gap junctions are when we have to provide a passageway between the insides of neighboring cells so that they can share electrolytes and other substances. In your heart, your heart muscle cells have to be able to all contract at the same time, and so they'll have a lot of gap junctions to share those ions. Desmosomes are there when you need cells to be held together strongly. It's kind of like stitches between cells, holding them together as strongly as possible. We don't want your skin cells popping loose from each other, right? So we have a lot of desmosomes there and some of these other layers, linings that you have in your body. Here's another version of them, of diagrams of them. This would be tight junctions over here. This would be a desmosome. And these would be gap junctions, forming passageways between neighboring cells, the insides of neighboring cells. Yeah. So those are examples of ways that proteins in the membrane can help to link neighboring cells to each other, forming membrane junctions. The other aspect of membranes and how they work is how membranes have to allow for transport of materials across the membrane. Stuff has to move into and out of the cell across the membrane. So they're working, they're dynamic. Things are going on in the membrane. They don't just sit there and act as a barrier. Sometimes things can get right across the membrane without the cell really having to do anything. That's called passive transport. It doesn't take any energy. The membrane doesn't have to work. It just lets things happen however it wants to happen. Diffusion is an example of passive transport. Diffusion is when you have an area of where a substance is highly concentrated and it just naturally spreads out and reaches equilibrium in whatever container it's in. Like if you take some food coloring, red food coloring particles, you drop it in a cup of water, that's what this is supposed to be on the screen. Those particles will eventually spread out. You don't have to stir it or anything. If it's just sitting there still, the particles will still eventually spread out and reach equilibrium because of diffusion. But we're not dealing with glasses of water, we're dealing with cells, right? And membranes. So when you're dealing with a membrane, here is the phospholipid bilayer right here. And sometimes you have a substance that can move right across the membrane. Now these are phospholipids, right? They're lipids. Sometimes you're dealing with things like steroid hormones. Steroids are lipids too. If it's a lipid, if they're both lipids, they can just kind of squeeze right in between those phospholipids and get across and eventually reach equilibrium. That's simple diffusion across the membrane. But sometimes there are things that are not made out of lipids, like some of the proteins or some of the ions, and they would come along and hit this membrane, and then they'd bounce right off. They wouldn't be able to get across. So if we needed to get those types of things, those other things across, we would have to create a passageway. We would have to facilitate their movement with something like a channel protein. That's why we call it facilitated diffusion. So here's a channel protein. It's like when I drew something back here. Where is it? It's a long way to go. There it is. Like this. This is something kind of like a channel protein. If it has a passageway, that lets something move through the passageway. Right? So if we go back to where I was, there we go. This is that channel protein that's letting something move through that hole that it has opened up and made across the membrane. So that's still it's still diffusion, it's still passive, but now that we've made a way for something to move. Osmosis is another type of passive transport, but it only involves the movement of water across the membrane. The standard experiment illustrating osmosis is you take a U-shaped glass tube or something like it, and you put a membrane, some material down in the bottom of it that will only let water across it. And you fill up water to equal levels on both sides of this tube. But in one side of it, you sprinkle in a little bit of salt down there, and in the other side, you pour in a bunch of salt. So there's a lot more mixed in on this side of the membrane than there is on the other side. That stuff, it wants to spread out. It wants to diffuse and reach equilibrium, and it wants to spread out so much that it ends up creating a little bit of negative pressure, sucking the water across that membrane. And you come back later, and there's more water on the side that has a lot more stuff dissolved in it. Our cells work that way too, our cell membranes. If we're dehydrated, the fluid outside of the cells is more highly concentrated in those solutes. And it ends up sucking water out of the cells, and if it happens too much, the cells can get kind of dried out and crinkled up, and they stop working, and you can die from dehydration that way. Parts of your body stop working before you kind of go into the death spiral of dehydration after it goes too far. Or if it's unbalanced the other way, then the cells can end up swelling up with water. Like if you drink way too much water, normally if you drink too much water, you just have to pee a whole lot. But if you drink water too fast for your urinary system to keep up with, it's called water poisoning. And that can make your cells swell up, and you can actually die from water poisoning that way. So if you ever get challenged to a drinking game with only water, don't do it. You can actually die from that just as easily as you could from alcohol poisoning. So, that's one of those homeostatic balancing acts. Our body is trying to maintain osmotic fluid balances all the time, usually through your urinary tract, urinary system. So those were types of passive transport. I also need to tell you about active transport at the membrane. Active transport mechanisms need energy. They're active. We're using ATP because we are trying to move a substance in the direction that it does not want to go, against its concentration gradient. In this case, we don't use channels anymore. We use pump proteins that use ATP to push substances into or out of cells. In this diagram, here's your simple diffusion across the membrane. Here's your facilitated diffusion with a channel protein. But here's our pump protein. This substance, if we're dealing with normal concentration gradients, if we're dealing with normal diffusion, this green stuff would naturally want to go that way and reach equilibrium by evening out. But this just appears to be something that this cell likes to have on the inside, so it's going to spend energy sucking more of that green stuff in as much as possible. Makes that sound all the time, sucking stuff in. All right, that's how pumps work. I always use this example. It's kind of easy here in the shoals where I'm recording this because we got the lake and the dam, and if we need to let water out of the dam from the high part to the low part. Let's see if my direction dam is that way. So from the high part to the low part, all we do is we open up some of those floodgates, those channels, and then the water goes however the water wants to go. That's facilitated diffusion. But if for some reason we needed to get the water up to the other side, we'd have to put in a whole bunch of pumps that would use electricity power to push the water up the other way. And so that's the idea between passive versus active transport. One more type of active transport is a little different. It uses things called vesicles. Vesicles are, think of them like little bubbles, sacs, made of phospholipids, just like the cell membrane, made of the same stuff, but they're just little bubbles of it that are for carrying clumps of whatever it is we're dealing with into or out of the cell. Maybe the cell has sat there and it's made something, whatever protein or substance is in here, and the cell needs to release it. So this bubble, this vesicle, will get carried. Some other proteins will help to carry it using energy. Will get carried close to the edge of the membrane. It'll eventually join up and bloop, it kicks out whatever it's carrying. White blood cells, if they are trying to absorb and eat and destroy bacterial cells, for example, they might surround it and draw it into the cell and basically just work the opposite way through endocytosis right there, forming a vesicle and taking something in. It still counts as active transport because it's still using ATP to move those vesicles around. That'll come up again in AP2 and a little bit in AP1. But for the most part, we'll be dealing with, I want you to make sure that you remember the difference between channel proteins and pump proteins. It's a very important aspect of how muscle cells work and nervous tissue cells work, neurons, to control our bodies. So to kind of sum up this information, I want to discuss very briefly the topic of a membrane potential. The idea here is that there's that picture again. The idea here is that because of this diffusion and the active transport of ions, charged atoms, mostly potassium and sodium and a little bit of chlorine and calcium, I guess, we end up building up an imbalance of those charged particles on the different sides of the membrane. This imbalance causes there to be a difference in charge, usually something like this. There's a negative charge on the inside, and there's an overall positive charge on the outside of the membrane. This leads to a difference in charges between the inner and outer side of the membrane. This difference in charge produces the potential for energy to be transferred, a membrane potential, or a voltage. You can measure it if you do it just right. It's like walking around on a carpet in winter. You know, if you're wearing some real fluffy socks and then you go over and touch the metal door, that friction of walking around on the carpet in the socks, it builds up a different charge in your body compared to a metal doorknob. And if those charges are different enough, you'll get your little static shock. That's energy being transferred. If we do that with our cells, we have the ability to transfer that really it's electrochemical energy in this case. So, to sum it up, the membranes of cells are charged negatively usually, which allows for things like nerve impulses and muscle contractions, which we discuss in modules 4 and 5, and that's why all this stuff that I've been talking about with the membranes is important for us. This is just a picture of a neuron. That's all it is. It was just there to fill up some space, but I thought it was a cool picture. We come back to this later. It's important for this class. It's important for you guys to know about. The rest of the cellular biology information I should be able to record a shorter video about, so I'm going to stop the membrane information here, and the last video for Module 1, we'll be discussing the cytoplasm and the nucleus of cells.